In this section of Chapter 24, we're going to be taking a look at certain mechanisms that can help preserve a species' identity. On the other hand, a postzygotic mechanism is one that may either prevent the survival of an interspecific or cross-species zygote, or may result in the infertility of such a hybrid. Geographic isolation, the first of the prezygotic barriers, can not only establish new species by way of allopatric speciation, but can also keep those two species genetically distinct by way of some formidable geographic barrier, whether it be a mountain, a canyon, a river, or even an ocean. Lions and tigers have different geographic ranges, and therefore they don't come into contact with one another at all. Tigers are indigenous to forest habitats, whereas the lion, the savanna. As a result, this lack of gene flow between both populations will keep each one genetically distinct, preserving their genetic uniqueness. If not for this geographic isolation, these two predatory cat populations would be able to interbreed and give rise to hybrids such as the liger, which we only observe in captivity. Isolation, another prezygotic barrier, occurs when two populations live in the same geographic range but do not come into contact with one another for mating purposes because of the fact that they occupy different habitats. An example, each one of these fly populations has a specific preference for laying their eggs either in apple trees or hawthorn trees. As a result, each one of these populations remains reproductively isolated from one another, even though the two trees may lie right next to each other. This reproductive isolation results in each population's gene pool becoming more genetically distinct from one another, thus preserving each species' genetic identity. Isolation is a prezygotic barrier whereby two populations living in the same geographic range and in the same habitats do not interbreed with one another because their mating seasons do not overlap. In this particular example, the height of the mating season for both the wood frog and the leopard frog differs. The wood frog's mating season reaches its height somewhere in mid-March, whereas the leopard frog's mating season reaches its height somewhere in early April. As a result, there is not much mating opportunities between individuals of these populations, and as a result, we would expect each population to remain genetically distinct from one another. Behavioral isolation is a prezygotic barrier that is important in areas where populations exist in the same geographic range, in the same habitat, and whose breeding seasons occur at the same time. Each population will remain genetically distinct because each population has its own unique courtship behaviors that will only be recognized by members within that population. Therefore, any crossbreeding between both populations is highly unlikely. Isolation is a prezygotic barrier that involves the incompatibility of individuals of different populations to physically mate with one another. In this example, anatomical structure of the flower parts of both the black sage and the white sage plant allow only specific pollinating insects to interact with them. As a result, this prevents any cross-pollination between the two populations due to these anatomical differences. Consequently, each population remains genetically distinct from one another. The last of the prezygotic barriers involves gametic isolation, which ensures that the egg cell of a particular species is only fertilized by sperm cells of the same species. Between egg cells and sperm of the same species is made possible due to the existence of certain receptor cells on the surface of the egg, whereby only sperm cells of the same species can interact. This is especially important among species that practice external fertilization, whereby it is desirable that only sperm cells of that same species are able to gain access to that egg. In contrast to prezygotic barriers, postzygotic barriers can either prevent a interspecific or cross-species zygote from surviving, or should it survive, it would probably result in the sterility of such a hybrid. To illustrate, the mating between a donkey, which has a diploid number of 62, and a horse, which has a diploid number of 64, would give rise to a mule, having a diploid number of 63. Mules are generally considered to be sterile, first of all, because their mating behaviors are incompatible with either the donkey or the horse. More importantly, its chromosomes cannot properly synapse during gamete formation. Look at the karyotype of a mule. We can see those chromosomes that are donated by the horse and the donkey. And we can see that, for the most part, these chromosomes are not compatible in terms of their number, their size, and of course their genetic content. This might be problematic during synapsis, which is necessary in order to produce viable gametes. As a result, mules tend to be sterile.